So welcome to everybody, and thanks for coming out to this month's edition of the CSMB virtual seminar series. We have two really great postdoc speakers that we're going to hear from today. But just before we get started, a few housekeeping things. So this webinar will be recorded, as you heard. And if you'd like to ask any questions, everybody's been muted, so you can use the raise hand function and we'll unmute you if you'd like to ask your question. Or you can also post your question in the chat and I'll read it out. I also want to welcome everybody on behalf of the CSMB. My name is Matt. I am the trainee board member representing the CSMB. And if you're interested in becoming a member, I highly recommend you join. We represent scientists across Canada, and there's many great perks of being a member. And you can check them out at this link down here if you want to know more. So now getting to our talks, we'll have our first talk today from Dr. Claire Martin. Claire is a postdoc in Dr. Anne-Claude Greengrass's group at the Lunenfeld-Tanenbaum Research Institute in Toronto, and she'll be telling us about dissecting time and space dynamics within the EGFR receptor signalosome. I'll let you share now, Claire.
So I forgot to unmute my audio for a sec. Perfect. I'm just getting Zoom out of my way. Great. So I just wanted to start off by thanking CSMB and the board for putting on this series. It's been great to connect with different people that I haven't met before, that a lot of whom I was able to meet at the CSMB annual general meeting, which was really fun and wonderful. And I'm excited to tell you more about my project, which is how proximity proteomics can eliminate spatiotemporal control of cell signals. And more specifically today, I'm going to be talking about the receptor tyrosine kinase, EGF. So EGFR is a receptor tyrosine kinase with important functions in development. It converts extracellular messages through a series of intracellular signaling events to direct cell growth, survival, and migration. In this simplified model, we can see that upon its activation by its ligand EGFR becomes tyrosine phosphorylated, which creates docking sites for these SH2-containing adapter proteins, including BREB2 and SHIC-A. These adapters act as scaffolds to recruit various proteins, including GAB1 and the RAS-GEF-SOF, which go on to potentiate MAP kinase and AKT signal activation. So this activation is all tempered by the action of the Sybil family of ubiquitin ligases, which mark EGFR for degradation following its endocytosis. So even though we know a lot about this way, which has been patched together over many years, there's still a lot unknown about many important things. So including while we view EGFR signaling happening in a static state on the plasma membrane, it's actually endocytosed, like many RTKs following its activation. And so we don't know a lot about the spatial organization of signaling events and how these signaling events are happening. So we don't know all the components yet. We don't know all the post-translational modifications. And also the temporal and spatial concepts. Further, we have only just begun understanding how these pathways are rewired during disease. So our lab uses proteomic-based strategies to help fill these gaps. So the classic approach to understanding protein-protein relationships is APMS. And so it involves biochemical purification of an epitope-tagged bait protein, which we express in an inducible manner at very low levels in live cells. So following gentle lysis, in order to try to maintain physical interactions, we pull out this bait along with any interacting proteins, which we then identify by spectrometry. So this has been a tremendously useful tool to study signaling by many groups. And a prominent example of this is from Tony Posson's group, who used APMS to look at signal regulation by the EGFR signaling scaffold SHIC-A. So by doing this in a time-resolved manner following EGF addition, we were able to see that waves of proteins are recruited in different kinetics, different time points following stimulation. And if we're able to then overlay post-translational modification data, such as phosphorylations of tyrosine, serine, and threonines, we're able to capture a systems level picture of how signaling works. So APMS works very well for soluble complexes, and temporal studies are possible if the interactions are stable and can be maintained through that purification process. It's also compatible with PTM mapping and quantification. However, there are some challenges that are inherent with APMS, including that non-soluble components are poorly profiled. So this includes things that are at membranes, such as receptor tyrosine kinases, as well as found at the cytoskeleton, chromatin, centrosomes. Further, cell lysis really disrupts the spatial context. So essentially, we have this bag of proteins that we're pulling out these complexes from, and we don't have a lot of information about where in the cell this is actually happening. So about 10 years ago, proximity tagging biotinylation strategies were introduced as a complementary approach to APMS. And it's really revolutionized how we're able to study protein relationships in living cells. And our lab is using the BioID flavor of this method. So in BioID, an abortive biotin ligase, such as mini-turbo, is fused to a protein of interest, and following the addition of cell-permeable biotin, the mini-turbo enzyme generates this cloud of reactive biotinyl AMP. This happens around the bait in about a 10 nanometer circumference, and these active biotins covalently bind to populous lysine residues near the protein. These are then captured by strep-avidin affinity, and again, by so APMS identification here is pulled from purification. So this method allows us to capture information on in situ protein organization without having to concern ourselves with maintaining the relationship post-lysis. And early on in its use within our lab, we really recognize that this approach actually transforms the bait into a sort of sensor that provides information not only on interactions, but also on where the bait might be residing in the cell, because it is capturing anything that's within a 10 nanometer distance. So this has the added benefit of separating the workflow so that we are able to capture these in situ relationships. It works on most organelles, even membraneless organelles, such like we would see in a signalosome. And nice for us, it works with the same computational pipelines that the Gingras lab, along with many others, have set up over the years. So we were able to use this approach in line with our current computational pipelines. We also have to remember, however, that this is a largish tag, an enzyme, and so we always have to check to make sure that our expression is neutral. What happens in both the APMS and BioID experiments is that we're left with this long list of hits, and untangling them really becomes our limitation. So when I started in the Gingras lab, my goal was to develop a pipeline to characterize protein relationships in signaling cascades. And I initially focused on that of EGFR. It was a good model for several reasons. We knew a lot about the pathway. So if we're going to be using a method, it's nice to benchmark a pipeline with something that we know a lot about. We know signaling is dynamic, meaning complexes are forming and falling apart over relatively short periods of time. So again, we can test our method's ability to capture these temporal parameters. And three, while we often use the signaling cascades as a static entity on the plasma membrane, we know that EGFR is being rapidly endocytosed and trafficking after its stimulation. So this really allows us to test our method's ability to capture spatial factors that are contributing to signal output. So to do this, I use kind of 4 BioID-based strategies. The first strategy is to capture temporal resolution of protein recruitment to and from the EGFR BioID bait. So using optimized 15-minute biotin windows, I performed BioID using C-terminally tagged EGFR mini-turbo, following its EGF activation over several hours. And what we were able to do is to capture waves of proteins coming to and from our EGFR bait that we were able to cluster based on shared profiles. So we saw 2 distinct patterns for proteins at the apical and basal cell junctions. And these were strongly associated with EGFR prior to stimulation and moved away following it. This differential patterning that we saw at each junction, we think might depict either distinct lateral movements of EGFR pools within membrane microdomains following its stimulation, or potentially preferential sites of EGFR in endocytosis. Proteins recruited to EGFR following its activation displayed 3 distinct patterns: those that rapidly associated and then dissociate with EGFR, making up signaling proteins that were dominated by ubiquitin modifiers; those that are recruited for a more prolonged period of time, which were more signaling proteins, as well as endocytic and trafficking proteins; and then those that appear to proteins that are likely involved in EGFR's retromer-mediated recycling back to the membrane for a period of time. So these profiles really reconstituted much of what we know about EGFR signaling, but it also identified new important components in these pathways, as well as provided us temporal information about these relationships. And even though it was illuminating, it still really remained unclear the pattern of events happening in that first 15 minutes, which, as you can see here, is where all the action is happening. And so we did further optimize the approach in order to provide 2-minute resolution of these signaling events. And so you can see here in 2-minute windows over the first 10 minutes, and then a window at 20 and 60 minutes, we can see that there's a much greater diversity in the kinetics of individual proteins than just what we were able to see with the 5 clusters in the longer periods. Okay, so now we have a pretty good understanding of the kinetics of, some 200 proteins that are differentially regulating EGFR, but we still lack information about how these proteins are organized amongst themselves. And so a multi-bait strategy is really helpful to gain information on spatial organization. And this is all based on the extent to which multiple bait prey profiles overlap. So in this method, we actually select a number of proteins identified as prey by an initial bait, and in my instance, it's EGFR. And then we perform reciprocal bio-ID profiling. We then use what is common and unique about their profiles to reconstruct basically of how the plays in a network are organized. So to date, I have performed this with 15 baits related to EGFR's initial prey profile. And at resting conditions, we do see that preys self-organize into signaling, endocytic, and trafficking kind of subgroups, but there's also a lot more connection than I would have expected. And following EGF stimulation, we actually see, somewhat of a flux of this network where endocytic proteins are identifying signaling proteins and vice versa quite frequently. So my results really seem to suggest that there might be this mechanism where signaling nodes are actually assembling at sites of endocytosis. So when I look further into the mechanism of this, I could really see that this transition from the no EGF to EGF related signaling network was really driven by a gain of relationship in a core group of about 50 of the 250 or so proteins identified in the network. And so we can see here that by limiting our view to only those preys that are highly interconnected, which I've identified as having 3 or more baits identifying them in the network, that the number of proteins in this core group really only changes by 4 proteins following stimulation. And this black guy here is EGFR, SHIC-A is another one of the proteins. And so really, we're not actually changing the highly interconnected nature we're not changing the number of proteins that are being recruited to that group, but what we are changing is the number of interactions, which are represented by edges. So even though we're only increasing by 4 new prey proteins being part of this core group, we're actually adding more than 100 interactions. And so this phenomenon might represent a mechanism for how signaling and endocytosis can actually happen so quickly following EGF stimulation. Within seconds, or minutes. And there seems to be this, loose scaffold of proteins existing at rest. And that's maybe how the cell is able to convert these proteins into a signaling node or an endocytic engine, because we're asking components to essentially add connections between neighbors that are separated by maybe one or two intermediate neighbors. So the last two strategies I use are conditional approaches that really help me to reveal the interdependence of these protein relationships. So in this simplified example, I have protein A and B maybe relying on protein C for recruitment to a bait of interest. And so if we were to knock out protein C, we would actually see in our prey profile not only a loss of protein C interactions, but also A and B. And similarly, if we were to mutate the bait so that the binding interface that's required for this, subcomplex to be recruited, we would see a similar profile. And in this manner, we can really tease apart linkages between components of a particular network using bioID. A really useful way to look at this, type of comparison, which involves really abundant changes of potentially hundreds of proteins at once, are these condition plots. So for my CRISPR knockout experiments, we compared EGFR bioID bait profiles again, but this time comparing scrambled control lines to knocked out of individual preys that I identified as EGF-dependent EGFR preys. And those preys that fall off the diagonal can really be identified as being linked in some way to that CRISPR target. So I've done this for now more than 30 EGF-dependent partners to test their impact, the network formation of the pool. And one of the key things that I found was actually a role for the EGFR adaptor protein GREB2 and broader network formation. And so here we can see, compared to scrambled, that knockout of GREB2 led to a staggering loss of proteins that through gene ontology, we identified as being important for not only signaling, but also endocytosis and trafficking. And this observation was not seen with other signaling proteins like Sybil or SHIC-A. So we know it's really specific to GREB2. And while we already knew that GREB2 is very important in EGFR signaling, this data suggests that GREB2 is particularly essential and that we don't see inherent redundant or adaptive mechanisms like we might. It further also resolves some specific protein relationships that GREB2 is actually essential for. And so that's really something that I've been interested in following up further. And so recently there's been some new interest into a role for GREB2 dimerization and its signaling. So this is a recent paper where the authors used a synthetic membrane in which they embedded the cytoplasmic domain of EGFR and added minimal kind of signaling pathway components to demonstrate that GREB2 dimerization is required for soft recruitment and activation. So what we can kind of, glean from this set of experiments is that they basically were able to show that GREB2 dimerization at this 160 tyrosine site is essential for this process to happen. What we weren't able to understand from this data was how this might play out in live cells. So with full-length EGFR and obviously much more complex pathway relays, if a similar result would have been found. And it's really exactly these types of questions that bioID does a really good job of helping us to kind of answer. And so I performed bioID this time using GREB2 as a bait, and consistent with kind of our CRISPR results. Here we see wild type GREB2's profile before and after EGF stimulation. We actually identify that a lot of the proteins that seem to have a GREB2 dependent recruitment to EGFR are indeed in GREB2's kind of own proximal profile. And then when we take the mutation that was shown in the previous paper and force GREB2 into a monoform formation, we do actually see quite a disruption in this this GAB SOS signaling profile. But it's not nearly as much as if we mutate GRB2's SH2 binding domain, which mediates recruitment of this complex to EGFR, or the SH3 binding domains, which we know is how SOS gets recruited. And so even though, this dimerization mechanism seems to be really, really essential in the artificial system, we see that the cell actually must have some compensatory amines that kind of dampen the effect in live cells. But what I found really interesting actually was even greater than the effect of SOS, we actually see quite a striking effect on the recruitment of ubiquitin signaling proteins, such as ubiquitin, ubiquinases, Sybil and Sybil B, and some of the deubiquitinases that I've identified in the profile. And that's something I'm really interested in following up on in the future. And it's kind of surprising that this GRB2 dimerization mechanism might have a role in this type of signaling. Okay, so hopefully I've been able to show you that by using bioID based strategies and bringing them together in a systems approach, we're able to improve our resolution of signaling. One thing I didn't touch on yet is something I'm really excited about, and it's we can actually capture phosphorylation of trays in our bioID profile. And so you can see here, these are all the phospho PTMs that I've been able to identify using my bioID strategy. And that's really important because not only are we capturing the spatiotemporal profile of these components using bioID, but we're actually able to capture the specific phosphorylated motifs that are mediating, a lot of these interactions. So I'm really excited to kind of expand this and overlay it onto my component analysis so that we can really get this full network picture of signaling. Okay, with the last couple of minutes, I just wanted to touch on something that's new for me to present. And so I did mention my bioID profiling has identified some new componentry. And the one that I've been most interested in following up on is this protein SPATA2, which is a ubiquitin binding protein. And so we can see here, using EGFR as a bait, you can see that following EGF stimulation, we do get this bump up in the identification of SPATA2 in EGFR's proximity. We also see this other protein, CYLD, which I'll touch on a bit in a sec. And so, we see this SPATA2 being recruited to EGFR in an EGF-dependent manner, but I also wanted to look at the reciprocal profiles that I did to try to understand a little bit better about, is it only being seen in relation to EGFR, or are we seeing SPATA2 in relation to any of the other components that might tell us a little bit more about where in the signalosome it's being recruited to. And so here I've profiled these other proteins by bioID. So this reciprocal profile, we can actually see both SPATA2 and CYLD quite robustly being identified within the signalosome. And so we're able to pretty confidently identify this new potential signaling partner through this method and have a pretty good idea not only its temporal profile, but also how it spatially is being organized. And so what does SPATA2 do? Well, it's actually quite well known that SPATA2 works with this deubiquitinase protein CYLD to perform lysine or K63 deubiquitination in cell signaling events. And so what we normally would have, a protein of interest that a ubiquitin ligase would ubiquitinate. Maybe this might be Sybil. And so then we have this, polyubiquitinated substrate. And to do the reverse, so remove the polyubiquitins, a deubiquitinase, which will have a USP domain, will remove the ubiquitins, creating, back to the starting state. And so one way it does this is by linking to ubiquitin binding domain containing proteins, of which SPATA2 is one. And so this, ubiquitin binding domain protein can act as a scaffold to bring substrates to a specific deubiquitinase or DUB. So CYLD is the DUB that SPATA2 likes to interact with and bring substrates to. And so I was really interested in understanding, more about what this could be doing in the pathway. One of the reasons I was interested is because when I was looking at my, novel hits, you could see that SPATA2 actually seems to have a significant role in survival probability in this case, specifically melanoma cancers. And so we can see here that with low SPATA2 expression, we have this striking drop in survival for melanoma patients. And so I wanted to see, how my bioID pipeline might help me understand why SPATA2 is so important and what it could be doing in the EGFR signalosome. I went back to my CRISPR bioID screen. And so here we have EGFR as our bioID base. And after knocking out either SPATA2 or CYLD, we could see that there was actually a modest but convincing bump in the recruitment of GAB1 receptors as well as PI3 kinase componentry to EGFR. That would suggest that the PI3 kinase AKT way might be altered in the absence of SPATA2 or CYLD. And that's indeed what we saw by Western blot. We actually see increased activation of EGFR in these HeLa cells with knockout of SPATA2 or CYLD. And this is associated with heightened phosphorylation of AKT and its activation. And so I think that following up on this data in melanoma lines, which I'm doing in collaboration with Ian Watson's group out of McGill, I'm really excited to maybe, convert my various systems network-based investigations, which all occurred in HeLa cells, to actually maybe understanding something a little bit better in melanoma. So with that, I'm going to finish and hopefully get some questions. Just wanting to highlight the funding that I've had for my fellowship, and also in bold all the people who directly have contributed to this. Thank you.
Awesome. Thank you, Claire. That was really impressive work and quite a lot of data you've collected. Do we have any questions from the audience? And feel free to raise your hand. Oh, there's one in the chat already. So the question is, have you found any cadherins or catenin recruited to EGFR?
I would have to go back to my data to confirm. I actually see them associated with EGFR prior to stimulation. And so very well in those very early seconds to minutes after stimulation, they could be affecting beta-catenin cadherin signaling, but I actually see them most strongly associated in that first and second group where EGFR seems to actually decrease in their proximity after stimulation. But I see all the classic beta-catenin and cadherin components.
Awesome. Any other questions? Feel free to raise your hand and I can unmute you if you'd like to ask it. There's nobody else from the audience. I have one more. I was really excited about your work studying PTMs with this, and I just had a more technical kind of question. Often you have to do enrichment for PTMs. With the bioID approach, are you just detecting these straight from your strep-evident pull downs, or do you do additional rounds of enrichments? Yeah, so that's what's so exciting. And this kind of stuff is actually kind of being led by another postdoc in the lab, Hugo Dion. But yeah, we are actually able to see it just in our straight pull downs, which is super exciting. So no need for PTM enrichment, nothing. They're just being identified as the peptides for the protein. So I'm super, super excited, which this has also been what's delaying me. Part of what's delaying me getting the paper together is that I think it's worthwhile to see how much further we can push this PTM in the bioID data. For sure. Yeah, that's really awesome. Okay, any other questions? Seeing none, I think we'll thank Claire for her really great talk, and then we'll move on to our next talk. So next up, we have Dr. Tina Singh from the University of California in Berkeley. She's currently working in Dr. Elchin Unal's lab, and today she'll tell us about leveraging gametogenesis-specific rejuvenation pathways to counteract cellular aging.
Great, thanks. Let me just get the presentation started. Okay, can you see those slides and hear me okay?
Don't see you sharing yet. There we go. Looks great.
Sharing, and you can hear me?
Perfect.
Okay, wonderful. Thank you. Well, thanks for the introduction. I'm going to start the talk off by letting you all know that I'm just getting over a cold, so apologies if I need to pause for a sip of water. I was really honored to be invited to give a talk at this seminar series as a fellow Canadian. And so I learned from Kathy when I was speaking to her that there's actually a lot of students on this call. So I thought before I jump into the science, I would take a moment to just tell you a bit about my trajectory to where I am now. I have been using budding yeast as a model organism for my whole career. And so I guess I'll start by saying I was born and raised in Sarnia, Ontario, which is a small town that's just an hour west of London. So that's where my journey began, which is my undergrad degree at Western University. Uh, I I learned about the awesome power of yeast genetics there. And so we use yeast as a model. This is the same yeast that we make beer and bread from. And I worked with Dr. Megan Davey and Dr. Chris Brandel to use budding yeast as a model organism to study DNA replication. I then moved to the University of Toronto, and I used budding yeast to study chromosome segregation in Grant Brown's lab, and I also used a lot of fancy robots to learn how to use yeast in a systematic way to do genome-wide screens. And so here's an example of us using those same robots to draw a picture of our supervisor. I then decided to make a big move for my postdoc to University of California, Berkeley, where I'm in the Department of Molecular and Cell Biology in Dr. Elchin Unal's lab. And I just wanted to mention that Elchin runs her lab jointly with her best friend, Dr. Gloria Brar. And so this is obviously a unique lab dynamic. Our labs are on paper independent, but they're run jointly. So we actually have two labs that have all of the same lab meetings, and I benefit from having two PIs as mentors. So if you're interested in learning more about that dynamic, I'm happy to tell you about it. And we affectionately call ourselves the Brune Lab. Okay, so today I'll be telling you about, oh, sorry, and I was going to say, I wanted to share my trajectory in case anyone on this call is interested in learning what it's like doing a postdoc in the US. I'm happy for people to contact me after the talk. So today I'll be telling you about my postdoc work, which is focused on trying to leverage these gametogenesis-specific rejuvenation pathways to counteract cellular aging in our favorite model organism, budding yeast. And the overall goal of my project is to try to revert cells from an older state back down to a younger state. So the aging field has collectively defined 9 hallmarks of aging that represent different kinds of damage that accumulate within a cell over time. And so as we learn about how these different types of damage accumulate and contribute to the decline of cellular health, we're entering this exciting new era where scientists are trying to find new strategies to combat or reverse these hallmarks of aging. So one strategy that scientists are taking is to look at nature and look at these scenarios where there are these organisms that seem to live longer than you might expect. And so if you live longer than expected, you get to be on the cover of a lot of these fancy journals. And so I just wanted to highlight a few of them just for interest's sake. So for example, the Greenland shark lives, I think now they're finding Greenland sharks that live over 400 years. And so, you know, of course, if we can understand how this is happening, maybe we can leverage some of these pathways to help humans. So in this case, it seems that these organisms actually just undergo development more slowly because their metabolism is extremely slow because they live in very, very cold water. And so these organisms actually don't reach sexual maturity until they're over 100. Another organism that's gained a lot of attention is the very attractive naked mole rat, which is about the size of a rat, but lives over 30 years. And so there's a lot of studies trying to understand how this rodent is able to live so long. And in part, one interesting tidbit I've learned is that they express this protein called hyaluronin that seems to have cytoprotective properties. Studies in dogs have revealed that, as many of you probably know, uh, large dogs tend to not live as long as small dogs. And this is due, at least in part, by the fact that small dogs have a different allele in genes that are important for the insulin signaling pathway. And so the reduced insulin signaling not only contributes to their small size, but also contributes to their longer lifespan. And then finally, this more recent finding from our collaborators at UC Berkeley was found by studying the rockfish that can live over 200 years. It turns out that these organisms seem to have increased levels of DNA damage response proteins, and they also express genes that reduce immune signaling when the organisms become very old, perhaps decreasing inflammaging, which is one contributing factor to decline in aging. Okay, so our lab takes a similar approach where we're trying to understand some natural pathways that prevent aging, but we actually study a pathway that exists in each and every one of us, which is pathways in our germline. And so have you ever considered from a human perspective that we all age and become adults, but our germline maintains the ability to produce these specialized cells, so either oocytes or sperm, that are able to fuse together to form a baby that is brand new and has its lifespan completely reset. And so our lab is interested in trying to understand how these cells are able to be passed on from generation to generation without passing on age. And so the way that we study these gametogenesis-specific rejuvenation pathways is we use budding yeast, as I mentioned. And this is because budding yeast actually also accumulates the same hallmarks of aging that our cells accumulate. So this makes it an excellent model for studying aging. And despite being a single cell eukaryotic cell, they actually can undergo meiosis by simply growing the diploid cells in starvation media. Okay, so let me tell you a bit about yeast aging. So yeast have a finite replicative lifespan. And so what this means is that a single mother cell can produce about 20 to 25 daughter cells before it dies. And this is because at the cellular level, the cells are accumulating a lot of age-associated damage. So if you compare a young and an old yeast cell, you can see, for example, that the mitochondria, which are tubular in nature in yeast, become fragmented as they lose membrane potential. The lysosome becomes deacidified and also fragments. The ER accumulates unfolded proteins. And inside the nucleus, different kinds of senescence factors accumulate, like protein aggregates, extrachromosomal circular DNA, and the nucleolus becomes very large and abnormal in shape. Okay, so as I mentioned, gametogenesis completely resets lifespan, and it does this in yeast as well. So if we allow a diploid yeast cell to undergo meiosis by growing in starvation media, the resulting gametes have their lifespan completely reset back to zero. So how does this rejuvenation work? Well, it turns out it's quite complicated. And so obviously this is a very busy diagram from a review we just wrote, but I'll just mention that our lab and others are busy trying to understand all of these different rejuvenation pathways that have to converge in order to ensure that none of the age-associated damage that exists in the precursor cell is passed on to the developing gametes. Now, one important player in gametogenesis rejuvenation is this master transcription factor NDT80. And so NDT80 is important for initiating a lot of the rejuvenation pathways. And excitingly, if you ectopically overexpress NDT80 in mitotically growing yeast, it's sufficient to extend lifespan by about 60%, suggesting that at least a subset of these rejuvenation pathways can be turned on outside of its natural context in order to combat age-related damage. So this brings me to my postdoc work, which was first to perform a genome-wide screen to identify all gametogenesis-specific genes that can extend replicative lifespan like NDT80, and then of course try to understand how these genes work in rejuvenation. Okay, so we know that rejuvenation occurs specifically in meiosis, and so we decided to do a genome-wide screen by using inducible yeast cDNA libraries. So let me tell you about how we made these libraries. So first we isolated mRNA from different stages of meiosis. And so we have 5 different libraries that include genes from the different stages listed at the top. So starting with this starvation library, this is technically a pre-meiosis library and includes all of the different genes that are turned on during starvation prior to meiotic entry. We then have the early meiosis library that includes all of the genes that are expressed during DNA replication and recombination events. We have a meiosis 1 and meiosis 2 library that includes all of the genes that are important for the first and second meiotic divisions. And then the late meiosis library that includes all of the genes that are important for gamete maturation. Okay, so we isolated these mRNA pools in duplicate and then did mRNA sequencing as a quality control measure. So in this heat map, I'm displaying the 2 replicates for the different libraries, so 1 and 2. And then the dark color represents high gene expression and white is low gene expression. And so just 2 patterns I want you to notice. First, if you compare replicates, you can see a high degree of similarity, suggesting that our isolation methods are reproducible. And then if you look between libraries, you can see these huge changes in gene expression that occur that are representative of the cascades of gene expression that are coordinated to have the ploidy undergo cell differentiation and then also cellular rejuvenation. As a quality control check, we also did some spot checking. So for example, we looked to see if there was a mitotic specific cyclin turned on. Of course, that should not be turned on at all in meiosis, so it's not present in any of the libraries. But we do see, for example, REC8, which is important for recombination, is on in the early meiosis library, as we would expect. And then NDT80, which is that meiotic transcription factor that I was telling you about, is important for the first and second divisions. So you can see that it's on it's present in the meiosis 1 and meiosis 2 libraries at the highest levels. Okay, so now that we know that our mRNA pools are of high quality, we converted the mRNA to cDNA using a highly processive reverse transcriptase, and then we moved the cDNAs into our destination vector of interest using gateway cloning. Now, this vector is a high copy number plasmid that has an inducible promoter. So for the yeast aficionados, it's a galactose-inducible promoter. And then we transformed our libraries into yeast using electroporation. Once we had our yeast libraries, we again paused for another QC check. And here we use DNA sequencing to look at the representation of the different meiotic genes that were present in each of our libraries. And so this is a 5-way Venn diagram where you can see each of the libraries are listed and the number of genes in that library in parentheses. And then you can see that there's a subset of genes that are unique to each of the libraries, many that are shared between libraries. And then we have 1,100 genes that are what we would call housekeeping genes, since they are present in all of the libraries. Altogether, this library represents over 5,500 genes, which is just over 84% of the annotated genes in the yeast genome. Okay, so we have our inducible libraries ready. And so let me tell you about how this screen works. So I'll remind you that the purpose of the screen is to overexpress meiotic genes to force rejuvenation to happen so that we can find new rejuvenation candidates that can combat cellular aging and extend lifespan. Okay, so how the screen works is you take an aliquot from one of the libraries, and we run each screen in parallel in either uninducing or inducing conditions where the gene is either off or on respectively. And we do every screen in triplicate. And each day we isolate old cells using this this technique called magnetic biotin sorting. So how magnetic biotin sorting works is that you actually biotinylate the original population of yeast. And so the biotin will label the outside of the yeast cell. Now, when we uh, allow these cells to divide, all of the cell wall and cell membrane is made completely de novo for the bud. So none of the biotin is ever passed on to the daughters. What this allows for is we can allow this original population to grow in a culture, and it will produce lots of daughters and age. And then we can actually pull down on the biotin to isolate those old cells again and use the old cells to inoculate a new culture to repeat this cycle over the course of 5 days, where we're continuing to age that original population older and older and older. So conceptually, then how the screen works is that early in the screen, the original population should be young and healthy and producing daughter cells. So you can see these cells each are expressing a different meiotic gene, and they're all producing daughters. So when I sample, you'll see representation of all of these genes in the population. Over time, we'll start to see cells die naturally of old age. And so when a cell dies, it will no longer produce daughters. And so this gene, this red gene, is going to disenrich from the population. However, by the end of the screen, you can see that there are still some cells that are able to produce daughter cells. And those cells contain a rejuvenation candidate, so a gene that seems to be allowing the cell to live longer than expected. And so we would identify this rejuvenation candidate using DNA sequencing. So altogether, our screens and old cells resulted in 125 genes that were enriched. And so these are our rejuvenation candidate list. And this is the 5-way Venn diagram showing you the overlap. So we did a little bit of refining. We removed different sets of genes that we thought were false positives. So for example, we took out any cells that were any genes that were also enriched in young cell screens that we did in parallel. So this removes any gene that might just be allowing the cells to divide faster. That would be another reason why they might enrich in this screen, and so would be a false positive. So we took those out. We took out genes that were involved in sugar metabolism since we're switching sugar sources to turn on gene expression. We took out some manually that were genes that were false positive because they overlapped with a gene that was a true hit. And then we didn't consider any of the gene hits that were in the starvation library only as meiosis specific, since they're not technically turned on in meiosis. And so this leads us to a list of 80 rejuvenation candidates that we're following up on. Now, before I tell you more about the rejuvenation candidates, I just wanted to take a moment and tell you about a couple of the hits we found in the starvation libraries, because if you're following any of the longevity science that's going on, one of the most tried and tested strategies for increasing lifespan is caloric restriction. And so we were reassured to find that we did pull out one gene, NMA1, that's part of the NAD biosynthesis pathway. And so this pathway already has a very well characterized connection to longevity. And so NAD plus levels decrease with age. And so increasing NAD plus levels will make cells behave in a younger state. And I just wanted to mention here that in a yeast model, overexpressing NMA1 can actually prevent protein aggregation in neurodegenerative models. And so in our screen, this is what the data will look like. We have the number of generations of the culture on the x-axis and then the gene abundance on the y-axis. So you can see when NMA1 was off, it's relatively stable in the population. However, when you turn it on, it enriches in the population. And of course, we want to validate that this actually recapitulates an extension of lifespan. And so we are using custom microfluidic chips to do single cell lifespan measurements of yeast overexpressing NMA1, and you can see a shift in the median lifespan. In addition to finding a gene that with known connection to longevity, we also found a new gene that doesn't have a previously characterized connection to longevity, TOS7, which has recently been shown to be important for the secretory pathway and cell wall organization, and has also been seen to localize to the lysosome. And so it's unknown how this gene is contributing to lifespan extension, but we do see enrichment in our screen and a shift in median lifespan. So this is good because it was a good sanity check to show that our screen is able to pull out genes that can extend lifespan. Okay, so going back to the 80 rejuvenation candidates that we found. If you recall the hallmarks of aging, what was really exciting is that many of these genes that were hits have known roles and pathways that might rejuvenate some of the hallmarks of aging. And we're excited about the diversity in this hit list because it might represent different pathways that have to converge in order to completely rejuvenate a cell. Some other interesting groups that we found 18 genes that are potential NDT80 targets. So that's that master transcription factor that I mentioned extends lifespan by 60%. And so we think that these may be the direct targets that contribute to this phenotype. 7 genes are part of the environmental stress response. So these are genes that seem to either turn on or turn off in all different kinds of stress. So perhaps these genes are helping the cells tolerate age-associated damage. 7 genes are important for transcription generally. And so, of course, it'll be interesting to see how the gene expression profiles change when we overexpress these genes. 27 genes have meiosis-specific transcript isoforms. So these are actually all truncations. And so we're interested in seeing how these genes affect longevity. Curiously, 15 genes have unknown function currently. And then I often get the question of how many genes have homologs. And so several of our genes have one or more homologs in worms or humans. And so if we sum the total number of homologs, there's 76 worm homologs and 35 human homologs that we could potentially follow up on. So in terms of future directions, as I mentioned, we're using these custom microfluidic chips to confirm lifespan extension in our hits. We'll probably start by seeing which subset can specifically rejuvenate the nucleolus, since NDT80 was shown to specifically rejuvenate the nucleolus. Of course, we won't stop there. We want to find genes that can rejuvenate other organelles within the cell, and ultimately try to develop gametogenesis-based strategies to revert cells from an older state back down to a younger state. So with that, I'd like to thank my supervisors, Elchin and Gloria, as well as a very talented undergrad that's been working with me, Michael, and my entire lab, past people that have worked on this project, our collaborators, and all of our funding sources. I'll end by thanking the organizers for inviting me and all of you for your attention, and I'd be happy to take questions. Thanks so much.
Awesome. Thank you, Tina. That was a really great talk. Another talk with tons of data, I think. You can just see how successful Canadian postdocs are from both of these talks. So are there any questions for Tina now? Again, like I said, you can raise your hand and I can unmute you, or you can type it in the chat. Maybe I'll start with one. So you have a lot of these hits, but surely as cells are being rejuvenated during gametogenesis, it's not just one gene that's driving the whole thing. So have you thought about trying some of these hits in combinations, maybe to identify either parallel pathways that don't really synergize, or different pathways that do synergize?
Yes, I think that that's a great point. Actually, one thing that's been happening as we've been doing the lifespan extensions is that a lot of these genes seem to be extending lifespan very modestly. And I think that exactly what you said, that combining some of these genes might help us reach that 60% that at least NDT80 is able to extend lifespan by. However, because the list is so diverse, I think another interesting aspect is if we can understand what aspects of the cell each of these genes are rejuvenating. If we take genes that, for example, rejuvenate the mitochondria and combine that with something that's rejuvenating the nucleolus together, can we extend lifespan even more? And how far can we push that limit in yeast?
Right.
Yeah,
That sounds
question.
awesome.
Thanks.
So there's one in the chat from Alexis. So she says, has NMA1 overexpression been tested in animal models? And by extension, is the mechanism of NMA1 loss in aging understood?
To my knowledge, no. And I think that in general, the NAD plus just pathway has been well characterized. So the idea is that as your cells age, the NAD plus levels decrease, but if you can increase them back to higher levels, then the cell has more energy, and so will behave more like a younger state. So I'll be honest with you, the easiest ways to have your cells increase their NAD plus levels are things that you probably already know, like exercise is one that is really, really good at increasing NAD plus levels. But there's like a lot of hype right now you know, in the science world, and also the media has like jumped on the NAD plus bandwagon of all of these NAD plus precursors that might help us increase the NAD plus levels without exercising. So the connection, I think, has been very well studied, but whether any of these supplements are going to have any positive effect is not known right now. I think that, you know, people are starting to study these pathways, but it's going to take a long time in the humans, but it definitely has been shown to work in animals. So I would say I don't know that the gene specifically, like overexpression of that gene, has been studied, but definitely taking precursors to increase the NAD plus pathway has been well connected. So I suspect that overexpression of NMA1 would have a similar mechanism.
Awesome. Great. Any other questions? Okay, seeing none, I think we'll thank both of our speakers for two really great talks. And I just want to say, so this was our last seminar for this season. We're going to take a short break for the summer, and then we'll be back in the fall. If anyone here is interested in giving a talk, feel free to reach out to us, and keep an eye out for the form that we're going to put out looking for other speakers. So thank you to everyone, and hope you have a good day.
Thanks, Matt.
Thanks, Tina. It was a really great talk.
Oh, thanks. Thanks for having me. Yeah, this is so awesome. I would love to be, am I like on the email list now since I signed up?
Yeah, we'll keep the email list running, so you'll see all the new
Okay.
speakers.
Yeah, great. Sounds good. And yeah, let me know if I can reach out to my network of people here if you want others from the West Coast.
Yeah, that would be great. When we get the form for soliciting new speakers, I'll send it to you. And if you want to circulate it around your network, that would be really great.
Sure. Yes, I'd be happy to. Okay.
Okay,
Let's
cool.
catch up soon.
Yeah, see you
Great. Take
later.
care.